Hi, and welcome back. In this section, we are going to start exploring the special senses. So let's go ahead and get started. First, we're going to explore the sense of smell, also known as olfaction. And so for this sense, we're going to involve somewhere between 10 and 20 million olfactory cells. And these are modified bipolar neurons. So this is the first time that we have talked about bipolar neurons. Well, we're going to be talking about them a lot as we learn about the special senses, because those are the only places we find bipolar neurons. And so let's put you in the right part of your body. Sorry. This is the undersurface of the frontal lobe. So we are at the ethmoid bone right here, specifically the cribriform plate that has all those foramina. And sitting right on the cribriform plate is the olfactory bulb. And the bipolar neurons extend between the upper nasal mucosa and the olfactory bulb, going through those foramina. And so what's going to happen is the odorant is going to dissolve in the mucousy coating in the upper nasal passageway, and it's going to be binding to receptors which are on these really long cilia of the neurons. And that's going to cause the neuron to depolarize, require some ATP, and then we will have synapses in the olfactory bulb, and their axons will continue through. So let's look at the picture on the right. At the bottom, we have the mucus in the nasal mucosa where the chemicals have dissolved and the long cilia of the bipolar neurons are located. From there, when they are stimulated, the message goes to the cell body, and from there through the axons. And what you'll notice with the axons is this actually is a convergent mechanism where multiple axons are going to travel through the same olfactory foramen and synapse to a neuron in the olfactory bulb. And so that gives us to our secondary neuron. And the axons of these secondary neurons will travel through the olfactory tract. So in this case, the bulb is acting exactly like a ganglion in the peripheral nervous system. So the secondary neurons, axons, are then going to go to the limbic system, and from the limbic system to the temporal lobe, which is where the olfactory part of the brain is located. And it has to go to the cerebral cortex for you to be conscious of the smell. Please note that for olfaction, we are not going to be involving the thalamus, and this is distinct from the other senses we'll be learning about. Now, as you age, these neurons become less sensitive, and so you have to have a higher sense. They also tend to die off with any type of minor head trauma. I think it's very easy to visualize these little tiny holes. They'll be very easy to rip the axons and tear them. Another interesting fact is that a decrease in sensation is now being recognized as an early sign of several different neurological diseases, such as Parkinson's and Alzheimer's, as well as a total loss of olfaction is found with many viral diseases, not just the coronavirus that causes COVID-19, but other ones as well. So let's look in a little bit more detail about the physiology of smell. As I said, that olfactory epithelium is way high in the superior region of the nasal cavity. So we're looking superior to the superior nasal concha. And the chemical molecules for odor dissolved in the mucus that's created by those epithelial cells. And while they're in the mucus, they're going to bind to specific proteins called odorant binding proteins. So the odorant, which is what a chemical from an odor is called, the odorant binds to a protein called an odorant binding protein. Now, the olfactory receptor cells, in other words, these bipolar neurons, they have receptors for those proteins, and they're going to stimulate them. Okay? And so this picture is showing the plasma membrane of those bipolar neurons. And only a few molecules need to bind. And this is going to involve a second messenger system. So just reviewing it again, when these odorant binding proteins with their attached odor molecules bind to their receptor, a G protein gets activated. Activated G protein stimulates adenylate cyclase to be activated, and activated adenylate cyclase causes ATP to form AMP, cyclic AMP, and cyclic AMP is the second messenger. And cyclic AMP is going to open cation channels, plural, as in sodium and calcium channels, as well as calcium gated channels, because the calcium influx for the sodium and calcium, that calcium is going to bind to a calcium channel on the inside, and that causes chloride to exit. And so this is going to give us depolarization, which is going to allow our local action potential to be generated, which brings us up to threshold, and then we have an action potential which can travel along the axon. And so there are like 2,000 different complexes that can happen because there's a multitude of odorants. So from there, we're going to continue. That secondary axon is going to continue to the various olfactory pathways. And so I'm going to talk about the three most important ones. The first one is the one that evolutionary wise is the oldest. So like more than 50,000 years ago, it developed going to the hypothalamus. So this is the one that controls whether or not you salivate at a smell, or you sneeze at like the smell of pepper, or you have gag reflexes to certain really noxious smells. And this will involve your reticular formation system in your brainstem. Less old, but still old, is the amygdala. And this is has a little bit of limbic system. The amygdala is part of the limbic system, sorry, is what I wanted to say. And so this is going to link certain smells with the emotions, like when you smell chocolate chip cookies, everybody always feels like, oh yeah, that puts me in a good mood, got rid of my funk for the day. And then the newest system is the system that takes us to the cerebral cortex, where we can consciously perceive and identify the smells. And this involves the insula, the deep part of the temporal lobe that we've talked about previously. Okay, so that's it for olfaction. So now let's move on to taste or gustation. So let's talk about some structures first. And those structures are papillae, not the taste buds, but papillae on your tongue. And we have 4 different types of papillae. The first are called circumvallate or vallate papilla. And if you pull out your tongue about 2/3 of the way back, you will see somewhere between 7, 10, 11 of these forming a backwards V in the back of your tongue. And they tend to stick up a little bit. They're quite prominent and easy to see on your tongue. All the other papilla are going to be anterior to that. The next one are called foliate papilla. And these are on the sides of your tongue, fairly posteriorly. And for adults, they look more like just wrinkles there. For children, they're much more leaf-like looking, which is how they got their name, foliate. The third type form all these visible bumps on the anterior 2/3 of your tongue, which look like little miniature mushrooms sticking up. And so they're called fungiform, which just means in a mushroom shape. Those are the 3 types of papillae that are going to have taste buds. And so they're very discreet and easy to see on your tongue. The 4th type of papilla is the one that you actually have the most of, okay? And it's called a filiform papilla. It looks kind of like the flame of a fire. And that's what gives the whole shagginess to the surface of your tongue, are these filiform papilla that do not have taste buds. So just refreshing your memory about things we have learned earlier this semester, there are 3 cranial nerves that are actually innervating the tongue for taste. The anterior 2/3 of the tongue is going to be the facial nerve. And then the posterior 1/3 of the tongue is going to be the glossopharyngeal tongue. Now, the vagus nerve does do some taste, but the taste it does is way back at where the root of your tongue attaches to your pharynx. So it does that part of your tongue, and then it does some pharyngeal structures surrounding that area. So if you think back to grade school, you learned about some basic tastes: sweet, sour, salty, bitter. Now we know that there are a multitude of different tastes, but there are 5 primary tastes. And you may have had to memorize, well, the tip of the tongue is where you taste sweet, and over here is where you taste salty, and then the bitter is tasted back there. And so, yeah, the tongue has been mapped, and there are certain areas for certain tastes. And so we're going to learn what parts of the tongue learn those 5 tastes. So look at them and make sure you understand. Yeah, the entire tongue can do all 5 of the tastes. Okay. So if you put salt anywhere on your tongue, you're going to be able to recognize that it's salty. It's not only one area of your tongue that deals with salt. So let's talk about these 5 basic tastes. Okay, so sweet, basically, we are looking at organic compounds, compounds containing carbons in them. So our sugars, as well as our fake sugars, our saccharins, our aspartames, all of those are going to activate our sweet taste buds. Salty has to do with metal compounds, and specifically we're talking sodium-based and potassium-based. For sours, we're looking at acids. So we're looking at hydrogen ions in lemon juice and vinegar, in wine gone bad, because wine gone bad becomes vinegar. And as opposed to acids, the alkaline substances, the alkaloids, those are what give us a bitter taste. And so it doesn't have to be something super bitter like lye soap, but we have alkaloids in our dark chocolates, nicotine, caffeine. So there are lots of substances which can give us a bitter taste. And then our newest well-recognized taste is umami. And umami is that meatiness. So it's the difference between the taste of your ramen soup with the package and not with the package, because the package contains glutamate, and glutamate and aspartame, those are amino acids which really give us this meaty taste. And so it doesn't require meat, it just requires these amino acids. And so things like mushrooms can really give you this umami taste as well. Now, we also know there are a lot of receptors there for things like our fatty acids, for some of our other metals, and for our watery taste, but we're going to stick with the big 5. Okay. And when you're young, you're much more sensitive to these tastes. And as you age, especially by the time you hit your 50s and 60s, the ability to distinguish between the tastes declines. So where are those taste buds? Well, the taste buds are on the side of those papillae. So let me go back to those papillae because I forgot to point them out here. And so, for instance, here we have big circumvallate papilla right next to the filiform. And the taste buds are these structures. They're not on the surface, they're on the sides of the papilla. Here they're on the sides of the foliate papilla. So that's why on this picture, I moved the epithelium to the direction it would be on the side of a papilla. And so the taste buds are interspersed along the normal non-keratinized stratified squamous mucosa that you expect to find in the tongue. So they are these paler areas that you see among the darker purple stratified squamous epithelium. So if we look at these up close, here's what they look like. You can see that there are different types of cells in there, and then it looks like it's puckering towards a little opening at the top. So Let's revert to a drawing so you clearly can see what's going on. So every taste bud contains around 50 cells, and there's three major types of cells in there. There's the group called the taste cells. These are the ones that are important. These are the ones that give you taste, thus their names. Yes, there are different ones. We don't need to know about that. So, and in addition, between the taste cells, we are going to have supporting cells, and then down at the bottom of the taste bud, you will have basal cells, because these are the cells which will undergo mitosis to give birth to these other cells. Now, these cells in your taste bud have a lifespan of only about 10 days. Okay. Now, how can you tell the taste cells? Well, they are structurally different than the others, plus they are the ones that are going to have the sensory dendrites. Let me put my glasses on so I can see. The sensory dendrites around the base of them. I just wanted to make sure they were there. So these light purple cells, those are the taste cells. And at the other end, we have long microvilli, which are extending towards that opening, the taste pore. So let me take this and make it even bigger so you can see it. Sensory dendrites at the base of the taste cell, and at the apical surface of the taste cell, these long microvilli. So what are the microvilli doing? Well, they're increasing the surface area, not for absorbing and secreting, but so we can have more receptors to be bound to. So the molecules which are dissolved in your saliva, do you see the little saliva making a little pool inside the taste bud, bathing those microvilli? That molecule is going to bind to its receptor on the microvillus. Okay. Every cell has multiple different types of receptors. It's not one cell doing the sweet and a different cell doing the umami. They're all on the same cell type. And so we would get an action potential, and then a sensory neuron would carry it to the cortex of the insula. And so the insula analyzes it, and the cortex integrates it and gives you the average taste. And you go, ooh, that chocolate's really bitter. I can't use that for baking unless I have something sweet added to it. All right, so let's dive into this physiology of taste a little bit deeper. So the tastins, that's what the taste molecules are called. So smell molecules, what odor and taste molecules are called tastins. So the tastins in the food are going to bind to their specific receptors. They do not have to bind to a protein first. They're just going to bind to the receptor. And then that receptor cell will be depolarized. And then when it's depolarized, we'll cause release of a neurotransmitter, which then travels along the sensory neuron. However, there are several different methods for this depolarization to happen. So let's explore this a little bit deeper. I'm going to put my glasses on just so I can see the picture so I can point to it a little bit better for you. So for sweet, bitter, and umami molecules, all three of these are going to use a substance called inositol triphosphate, and you can just go with IP3. I for inositol and 3 phosphates, the P3. And IP3 is a second messenger. So let me tell you how we get to the second messenger and what this second messenger does. So the sweet, bitter, or umami molecule, this particular picture, it's showing a bitter molecule, binds to its receptor. When it binds to a receptor, that causes an enzyme called phospholipase. Well, it activates the G protein, first. And then the activated G protein will then activate an enzyme, in this case, phospholipase C, which is abbreviated PLC on the picture. And PLC takes a compound known as PIP, which is phosphatidyl inonosol biphosphate. Phosphatidyl for the P, inositol for the I, and the biphosphate for the P. So it makes PIP convert. It actually breaks it up into 2 compounds. One compound it breaks it up to is called DAG, or DAG, which is a diacylglycerol. And DAG actually, which isn't shown in this picture, DAG goes and it is bound to the plasma membrane. And DAG is a second messenger in its own right. It's just not involved in this system, which is why it's not being shown. So when the phospholipase C works on PIP, not only does it make DAG, it also makes our IP3, our ionosyl triphosphate. And this is a soluble molecule, so it's not going to be attached to the plasma membrane. What it's going to do is to diffuse away from the plasma membrane, and there is a receptor for IP3 on the membrane of smooth ER. So when IP3 binds to its receptor on the smooth endoplasmic reticulum, that's going to cause the sequestered calcium to exit, thereby raising the cytosolic calcium. Okay. And this is going to, that increase in calcium in the cytosol will then cause vesicles filled with neurotransmitters in these taste cells to release the neurotransmitters, and then those neurotransmitters will cause depolarization of the sensory neuron. Okay, so sweet, bitter, and umami all use IP3 as their second messenger, which means calcium triggering the neuronal depolarization. In addition to IP3, sweet molecules will also use cyclic AMP as their second messenger. So I'll just go over this super quick. We've had this many times. So as you recall, when the sweet molecule binds to its receptor, it's going to activate the G protein, which then will activate the adenylate cyclase, which changes ATP into a cyclic AMP. It hydrolyzes it, so we get a cyclic AMP and an inorganic phosphate. So the cyclic AMP then is going to activate protein kinase, and this kinase does what all kinases do, which is phosphorylate, and what it's going to do is phosphorylate a calcium channel in the plasma membrane of the taste cell. When that potassium channel, I'm sorry, I think I said calcium channel, it phosphorylates a potassium channel. And when that potassium channel is phosphorylated, it closes it so calcium can no longer exit the cell. And that is going to cause depolarization of the taste cell. That depolarization is then going to open a voltage-gated calcium channel, thereby allowing an influx of calcium into the taste cell, which then triggers the release of the neurotransmitters, and that will then cause the neuronal depolarization. All right, so we've got sweet, bitter, and umami covered. So let's look at the other two tastes, neither of which involve a second messenger. For sour molecules, remember, sour is looking at hydrogen ions in compounds, and so those hydrogen ions themselves will be binding to a potassium channel. And when hydrogen ions bind to a potassium channel, that means potassium can't exit the cell, thereby causing depolarization of that cell. Once again, opening those voltage-gated calcium channels, triggering the release of the neurotransmitter and depolarization of the neuron. So that's how sour works with its hydrogen ions. Salty molecules also do not involve a second messenger, but they work in a slightly different way. They are going to bind to sodium channels. And in fact, they're not really going to bind to sodium channels. They are going to enter through sodium channels and thereby causing depolarization, allowing calcium to enter voltage-gated calcium channels and releasing neurotransmitters for depolarization. Okay, so hopefully you understood the physiology. So let's talk about the pathway. So all of those taste cells were activated by the 5 different types of taste, and what they all did was they caused an action potential to be generated in the primary sensory neuron, whether it's the facial nerve or the glossopharyngeal or the vagus nerve. And so all three of those primary neurons then will go to the medulla oblongata. As you know, they're all attached to the medulla oblongata, so that's where they're going to go to synapse. And so the activation of these primary neurons is what can trigger some reflexes, like for instance, the salivation, the gastric juices, if I'm sure you all learned about Pavlov's dog previously, possible gagging and possible vomiting. So now we are going to have cell bodies of our secondary neuron in our medulla oblongata, and then their axons are going to go to the thalamus, where they are going to synapse. So we're finally involving the thalamus, which is our primary sensory relay center. And from the thalamus, we will then have tertiary neurons, which will travel all the way to the insula, and they have to go to the cortex for you to consciously perceive what the taste was. So, looking at taste, the temperature of the food that you're consuming affects the taste buds and the depolarization, as well as the texture of the food, which explains why some people really don't like mushy foods and some people don't like eating cold pizza, whereas other people are fine with cold pizza. And the other thing you really need to realize and remember, and if you had thought about it, you would have agreed with it, is that the vast majority of your taste clues are actually coming from your nose, from the sense of smell. And you all know this because when you have a cold and your nose is all stuffed up and you can't smell anything, you tend to lose your appetite because you can't really taste anything. All right. Now, these taste cells, these taste buds, even though they tend to be on the sides of the papilla, they are still susceptible to trauma. For instance, if you drink super hot coffee, the hot coffee can damage the cells, okay? So if all you're doing is damaging them, like for instance, a hot beverage, the taste can return in hours or maybe in a day or two. But if it's something like you're eating super hot, greasy pizza and the hot grease goes on, then it's a lot harder for your saliva to wash the grease off, and you're much more likely to actually kill that taste cell. And if the cells are killed, it can take 7 to 10 days for them to be replaced by those basal cells. And so you're gonna have funky taste for 10 days to 2 weeks, okay? And with this, I am done with olfactory and gustation, and I will see you again soon as we continue our exploration into the special senses. Thank you for all your hard work and have a great day.